So, hello everyone again. I'm Oliver, and I want to welcome you again to another Saturday microscopy live stream. Yeah, as always, I have to get some stuff organized here. Yes, here we are again, and welcome today. I posted a question, and I was collecting some requests on things to put under the microscope, and indeed, I did find most of the things that you requested. So we'll have a look at those things. Here's already a pond water sample. And usually the first 2 or 3 minutes, as always, I want to say hello around the world. Gives people time to join into the live stream. Yeah, and so basically, hello from Ireland, from Canada, from the United States. Okay, from the United States again, from Wales. Yeah, it's quite close for where I am. London, UK. Sound and vision is good, thank you. Northern France, Netherlands, United States, Athens in Greece. Very nice. Oh, I basically New Jersey, Switzerland. Okay. Okay. Poland, Finland, London, UK. So hi, and I'm happy that you were able to join. You are able to join me again. I hope that you had some the last couple of days that you had some nice Christmas holidays. Maybe some of you are still in holidays here. Hello from Australia, Vietnam. Okay, yeah, I'm totally global. That's one of the things that I like about this. That's yeah, microscopy, nature observation. Yeah, it's. I like to have a community all over the world. So very briefly of what I want to do today is, as I mentioned, I already. Excuse me, I have to cough now. So here we go. So basically, I tried something different. I basically posted a question on what do you want to observe, and one of the first comments in this chat, and if you scroll all the way to the top, there was a request to put in essentially some aquatic life and algae in a winter pond. So as a matter of fact, a few hours ago, yeah, we had nice weather here in Austria. I'm in Austria in Central Europe. Today's weather was quite nice, so I visited a local pond and I collected this water sample here. Look, it was not an easy water sample to collect because there were not many algae and not many water plants. But if you just look at it carefully, there is actually a little bit here on the side. Yeah, there is something, some larvae or so that were moving around. So I'll put this under the microscope. Number two, there has also been another request to put some more crystals under the microscope. So I prepared some citric acid. Last week I did some vitamin C. Today some citric acid. Just 4 minutes before the start of the live stream, somebody requested that I put some washing water from a washing machine. So I also collected that here. Yeah, I've got over here also. So from last time, these were basically, I was trying to grow my paramecia here. I don't know, this does not look very nice. So I'll have a look at this here as well. Okay, so lots of random stuff today. And yeah, we're gonna see how this we're able to go today. In other words, okay. I also have over here my the coffee warmer again. Yeah. By the way, you see that basically I made the picture a little bit larger here as well. So I don't even. We're just gonna get started, I would say, okay, because we've many things to cover today. And what I would like to do now is I would like to show you just a minute and a half a video from the pond that I visited a few hours ago, where I collected my water sample, with the one that you see over here. Okay, and maybe for those of you, and I know that there are a few people who are also from the area where I am, so maybe you are able to recognize this pond. So I'm gonna just start the video. There is no sound. Okay, that's the pond, and it was actually yeah. So the thing here is the pond. That's basically, yeah, you can see that it was actually a little bit frozen. Yeah, and yeah, the stage view. Yeah, I still have the stage view. Don't worry about that. It's just the desktop view where I made my face larger. You can see that over here, yeah, there was some snow here as well. Some ducks because yeah, there was actually an area where there was no ice. Okay, and you can see over here that's the shore. Basically, yeah, not a lot of green stuff here, right? It's a lake or pond. It's an artificial one, okay? So just for your information, this one was actually, I think back in the 1960s or so. Yeah, that's what I found. Yeah, that's the only thing that, almost the only thing that I found here. So back in the 1960s, it was actually used to gather gravel. Yeah, and so this is not a natural pond. Yeah, I wonder who this guy over here is. And it's not a natural pond, and now it's used as a recreational area. And for those of you who are also yeah, from the region, yeah, this is the so-called the Pichlinger See. Yeah, near where I'm living, right? And this is basically, I tried to collect some of the stuff here. Did not find a lot, okay? But I can assure you there is some living stuff in the water sample over here, okay? Yeah, it's a gravel pit, indeed. From hearsay, I think they used the gravel to make a local hydropower plant. Somebody told that to me, right? So they needed the gravel to make concrete, and so it's, I think it goes back to the 1960s or so. Now it's a nice recreational area. There were actually lots of people there today walking around the lake. Did not make a video of those, of course, but essentially, yeah, it was a nice afternoon that we had there. Yeah. So yeah, Santa Oliver with nice crystal presents. Yeah, you will be surprised which crystals I'm going to present to you. I've got a special crystal preparation for you today, which I did not mention yet. It's not only citric acid. There's another one as well that I'm going to show you. Yeah, seriously. And if you, yeah, it's an interesting one that I'm going to show you. Yeah, I'm going to make a crystal preparation that yeah, that you have not seen yet before, maybe. So yeah, so what I'm going to do is as always, just for your information, this this water plant over here is Ceratophyllum, I think. It's also commonly used in aquariums. And the important thing is when you collect water samples, of course, you are not interested in the water. You have to collect solid material. So some stuff from the ground, some rock samples, some decaying, decomposing leaves, whatever. It's got to be solid. You're rarely able to find interesting stuff just in the water itself. Okay, so the thing is the following. I don't want to destroy it either too much because I actually kind of hope to be able to grow it a little bit. So I need to put on my reading glasses here as well. So what I'm going to do is the following. Very simple. You take scissors, if I find them. Where did I put my scissors? Oh, man. Yeah, gee. Yeah, over here they are. Here they are. And I'm just going to put some of the salt. So I did not put this yet under the microscope, okay? I really hope to be able to find, usually there are some, I don't know, I've already in the past found some stentors, some vorticella, whatever, yeah, on here. And so we're going to see, and there is plenty of other stuff in here as well. And I can reassure you, I was able to find not a lot, but a little bit, which kind of shows that, yeah, even during winter time, there is quite a bit of life present, right? So cover glass goes on top. Again, I have not observed this water plant yet. It's too thick, as you can see. But you know what? Who cares? I'm just going to try it like this. And we're just going to put several samples yeah, under the microscope. And this is going to be, for those of you who don't know the format, yeah, this is highly experimental. So sometimes I don't even know what to expect myself. In most cases, as a matter of fact. So I deliberately try to, yeah, keep the tension up this way. So here is the. I need to turn up the light here. Here we go. Let's go down with the magnification first. It's not even in focus. Here we go. Yeah. Let me flip out the condenser here. Let me take this away. So this is now in regular bright field. So, and that's what we see here. I mean, there is a little bit of movement going on here, but I need to probably go up with the magnification a little bit. Okay. Yep. You see, there is also, oh yeah. Well, there is something here. We already found something. Yep. it looks a little bit like Vorticella maybe. See, with the condenser, it's so much brighter. Yeah, plenty of diatoms as well. I don't know what this is. Yeah, it's stalked. Yeah, so that's Vorticella. So basically, the bottom line is that you have to put some solid material under the microscope because on the solid surface you find microorganisms growing. Yeah. Yeah. So that is you can see some gliding diatoms as well. Yeah, to Vorticellae. Yeah, I think the problem here is a little bit that those water plants, I should have spread it apart a little bit to be able to see it better. But now I already, yeah, here too dark. Here is another one. Yeah, so Vorticella seems to look. Yeah, there are several of those. Yeah, but you see, they're all. Yeah, that's a nice colony of Vorticella here, but unfortunately not very nicely visible because yeah, it's kind of overlapping. Yeah, so for those of you who are a little bit new to the whole thing here, yeah, those here, these are basically stalked ciliates, and what they have is they have a stalk, and when they're disturbed, they're able to retract very quickly. Yeah. So let me go down a little bit with the magnification. Let's see a little bit more. Yeah. And also very nice. Ooh, did you just see this? Of course. Did you just see this? Where do these bubbles come from? Well, the water plant is doing photosynthesis, of course, and is producing oxygen, and that's why you can every now and then see oxygen bubbles appearing. Yeah, because of the lamp of the microscope, of course, this provides all of the energy. Yeah. Would winter have certain species which adapt to the cold and then bloom because of all the space and then get eaten by spring starters? Generally, I mean, it's normal that there's always some kind of a progression of organisms. So, of course, because temperature is super important, obviously. So, of course, there is a certain progression. You see. Yeah, bubbles appearing. So I wouldn't be surprised by that. But generally, during summertime, there is way more that you're able to see. There is lots of decomposition going on now during winter time because all of the plant material is kind of being broken down. So yeah, not so much primary production as we call it with photosynthesis happening. Yeah. Yeah. See. So I'm just looking for. Stuff here. Oh, here on the side. Yeah, you see everywhere where it's cut open. Yeah, the bubbles start to appear. Here as well. But again, over here, look. Here it's a little too bright. Here we go. Just a second. I'm gonna put this in here. See it much better. That is also a Vorticella over here. Here we go. There's something over here as well. Yeah, that's actually much better. Oh, there are 3 of them. Focus this. Yes, that's actually quite pretty. Okay, let's go up with the magnification yet more. I always have to turn up the light a little bit. Yeah, so these are basically 4 Vorticellae. Yeah. So, yeah. I would say, yeah, they're. Yeah, it's sometimes you also see so-called stentors. Usually, they're also attached to surfaces. Yeah, so quite nice. Yeah. Of course, you have a ring of cilia here, on the top, and the stalk, which is able to retract very quickly. Yeah, nice to see something moving again on the channel. Yes, yeah. So. That's a little bit. Sometimes that's what I like to do. Sometimes I just like to observe them. Nothing special going on. But it's actually, by the way, I did a little bit of research on those critters, and it's actually one of the fastest movements known in biology. Is this retraction of that in Vorticella? It's one of the fastest movements. I'm always kind of surprised a little bit that of the forces that are there. Yeah. Sometimes it depends a little bit. Sometimes you are able to induce the contraction or the retraction by tapping the slide, because every time when they're disturbed a little bit, then sometimes they pull together very quickly. I'm going to try that. I'm going to quickly try to tap the, no, not here, but if I tap, for example, the, yep, you see that? If I tap the cover glass, yep. Yeah, when I tap the cover glass, they quickly retract. Yeah. Yeah. 60 to 90 millimeters per second retract speeds, yes. But I mean, that's so, per second. Yeah, and that's considering that these are microscopic. It's a very fast speed. Yeah? So let's move on a little bit. Let's go down with the magnification, and then we're gonna look at some other things in here as well. I did collect some also decomposing material as well, but yeah, this one looks actually quite okay. I should have probably spread them apart a little bit more. You know what? I'm just gonna try that. I'm just going to try to. I'm just gonna try to spread it apart. Let me put the desk view again here. I'm gonna take out the slide. I'm going to carefully remove the cover glass again, and yeah, let's try to. See? Ah, yeah. You see, it's over here. Of course, every time when you do things like this, you might end up damaging the specimen somehow. Yeah. So, but a little bit, I guess, is okay. Cover glass goes again on top. And you know what I'm going to do now is I'm going to add a little bit more water here as well. Because then everything's... And then I'm going to have a look at a few... Oh, wow, okay. A few other things here. Sometimes when I usually it's like this, that when I prepare for the live stream, one of the most difficult things is always to think of a little bit of a topic, because more or less everything has already been done before. But usually when you try it again, usually there's always some kind of a variation. So I'm always how do you say, thankful for new recommendations. So this is now basically, I've spread it apart now. So let's hope, yeah, that we're able to see this. Look, yeah, wow, cool, okay. What I'm gonna do now is the following. I've got DIC, as you probably know, that gives it so much color. I'm going to just remove the DIC now, so that we're seeing it in bright field like most of you would be able to see it. So these are now the natural colors, okay? Ah, yeah, you've got over here some diatoms also on stalks, and sometimes, but unfortunately, no stentors. And some years ago, I was also able to find a hydra, which is actually quite nice. See, that's one of the problems, that they are kind of fast. Yeah. And we just keep on scanning the whole thing here. Yeah, and usually it's another thing. If you see somewhere some kind of a rotation or something spinning around, and that so means that there, of course, usually is some kind of a ciliate, like a vorticella next to it. It's actually causing this water movement. Yeah. So look at this. Okay. Yeah, and of course over here something that we should not ignore are all of those beautiful diatoms. Many of them are also moving and gliding around. Yeah. Let's go now yet further. Yeah. It's the beauty of nature. Yeah, and while we look at this, theoretically, I could slowly start to look at all of those diatoms here. I could start to have a look at yeah, maybe another part of the water sample, and occasionally I also read the comments here. Oh, that's also a pretty nice one over here. Yeah. Something that I also recommend if, yeah, I'm just reading here. Have you ever thought about visiting a reef aquarium store? Oh, that's interesting. I used to have an aquarium myself many years ago, a freshwater aquarium with fish. This was, however, long before I started this channel, microscopy channel. And right now, what I'm doing is I'm kind of keeping my water samples in a jar. But of course, I found out that essentially, very often the water samples in jars, even though they have microorganisms, usually the biodiversity is, of course, not quite as large as yeah, in nature. Reef aquariums are especially diverse. So this means I'd have to look if there is some kind of somewhere a reef aquariums that's a marine aquarium, so saltwater. I'd have to check if yeah, the local aquarium shops actually also do saltwater, marine water. Because I do know a couple of aquarium shops around. However, they're mostly all freshwater. Okay. Are those globs of green cells algae? So basically those that you see here could also be some kind of debris and some stuff of plant material that's being decomposed. Normally when you see algae, then you can actually see the cellular structure quite nicely. But these blobs, either these are several cells clustered together and stuck together, or they're just basically decomposing plant material. Yeah. The algae, as you see them, actually look pretty regular. Yeah? Like, for example, those blobs over here. Very often, the diatoms, for example, they make this slimy mucilage type of layer, and then things start to stick together. It's all part of the so-called the biofilm. Yeah. Yeah, so you know what I'm going to do is I'm just going to move on, and I'm going to put another thing yeah, under the microscope now. I'm going to try to find another specimen. Let me go back to the desk view here, and I'm just going to show you now a second one. I tried this also before, which kind of worked nicely. So I have to grab myself now one of these here, one of these things in here, stuff like this. Down here, where is this? If you take something like this, for example, this is way too much and way too thick. However, what you can do is the following. This is pretty rough what I'm doing right now. It's just pretty, but it actually does work. Just dip it here like this. And you kind of hope for the best, and you hope that things are being washed out. Okay, there seems to be already quite a bit of stuff here. Rest goes back, and yeah, I know this is a little bit messy. Maybe you can already see that there is yeah, it's kind of it's not completely clear anymore, but there is some stuff on here. So let's give that a try. I need to look for my cover glasses and always move the jars away, because otherwise if you tip them over, yeah. Can you enrich the sample by centrifugation? Yes, I can do that. But it's often not necessary. The reason is that if you just dip it like this, then very often, I tell you other way. I'll answer differently. Centrifugation is very good if you have, for example, pond water samples that are already very green. So you have a lot of algae and stuff floating around directly in the water itself that you want to have concentrated. So basically, swimming around. What I'm doing right now is I'm collecting or showing mostly things that are kind of on the surface of leaves and decomposing matter. Right, so it's actually a different one. If I were to take the clear pond water over here and centrifuge it, there would not be a lot there, because most of the things actually sink to the ground. Let's have a look at this one here. I always have to flip over, and let's have a look. So let's lowest magnification first. I need to flip out the condenser. Yeah, and that's how it looks like. Okay. Yeah, so we see over here a nematode worm. yeah, plenty of other things as well moving around. Sometimes a little too fast, I think. And yeah, let's have a look. I don't know. We're just going to increase the magnification a little bit. These things that swim around, they look a little bit like, also like Vorticella without a stalk. Here there are a couple of Vorticella with a stalk. So let's, there seem to be plenty of Vorticella here. Here again, look. Yeah. So here, wow, they have a huge stalk. Look at this. They have a huge stalk. And you know what? I'm going to make them contract again by tapping the, oh, they're contracted already. Yeah. Let's go up further with the magnification. I mean, look at this. It's actually one of the longest ones that I've ever seen. Look, they're huge. They're single celled, interestingly. Basically, it's yeah, if that's one cell over here, and there are these tiny here, the cilia, which are kind of moving the water, and then they're sucking in the food, and then they're digesting it in here. Huh? So these are pretty huge and long stalks, yeah. Yeah. So yeah, let's have a look. It goes all the way up. Yeah. Look how the stalk spirals up. Yeah. Let's. This is now 20 times. I mean, you're able to follow it. Yeah. Let's go up to 40 times. So I'm going to just stay with bright field right now, so that you're able to kind of because people were kind of wondering how I got emails, and they were kind of wondering whether basically what I'm showing is actually how you really see it, and I just therefore decided, okay, why not just stay a little bit also with bright field. Yeah. Oh, interesting. They here don't seem to be beating. The cilia don't seem to be beating. Now they started beating. Oh, okay, that's cool. Yeah. Yeah. So I think it's kind of cool. Huh? Feed the Vorticella and watch them long enough so that you can see them divide. Yeah, I've actually, this is actually also something that once happened. I was doing an enrichment culture, was adding a little bit of food, and then all of a sudden I had a huge colony of Vorticella growing there as well. Yeah. So I'm going to switch down the magnification again. Maybe we're able to find something else. Okay, here we go. What is this? Okay, those guys that you see swimming around, sometimes these are also Vorticella without a stalk. So yeah, when they're kind of dividing and reproducing, then they divide and then they swim off without a stalk. And this here is Pediastrum, I think. Oh, look at this! Haha! Look, that's yeah, water crustacean. Let's have a look at this one later on a little bit more. But this is something that I think, I don't know, is it Pediastrum? I don't know. That's a colonial algae. Could be. Yeah. So that is no, I don't. Yeah, this was some kind of a just a second here. Yeah, that's a colonial algae here. What do we have over here? Yeah. So you can also see the individual cells here. Yeah. So let's go up yet one more. Let's see if I'm able. Yeah. So that is the. So that's kind of nice. Yeah, so basically just what I want to say is, yeah, you take a little sample and then just observe it. So, but let's try to find this crustacean, whatever this was. Yeah, I wonder what this is thing here is. I've seen those already. It looks almost like an egg or of some sort. Yeah. You need to go down here with the magnification again. Wherever it, yeah, the diatoms moving around. Plenty of diatoms. Yeah, I don't know, can't find it anymore. But I want to find it. This means yet lower magnification. And okay, those are statoblasts of bryozoans. Okay. Yeah, and wherever it is, it should actually be large enough so for me to be also able to see directly on the slide. So I'm just quickly gonna go through it again. I don't know. Wherever it is. I'm going to just check a little bit the time as well. Yeah, it's already half an hour. It was this. Yeah. Here it is. Here it is again. So. Yep. Yeah, so in other words, I think what can we learn from this? Even in wintertime, when the lake is is half frozen, we're still able to find lots of living things. Oh, here, there's another colonial algae. Look at this. Here it is. Very nice. Yep. Yep. Here we go. This one is a little bit. Here we go. Yeah, it's quite nice. Yeah. Occasionally, if I'm not saying anything, just for your info, I'm just reading the comments. Yeah, as well. So okay, I don't know. Maybe the last very quick look is the following. I also collected a little bit of sand from the bottom. Okay, so because usually there is also, let me take this away again, and I'm just going to also show you a little bit of sand from the bottom here as well, and occasionally there are also, I don't know, I dropped it again. So here, and I'll just use the same slide. I'm just going to put it here next. And of course, sand is a problem because it's so extremely thick. So a cover glass is usually not the ideal thing here. So what I usually do with sand is I just directly observe it using the low power objective without a cover glass, simply to get a little bit of an overview of what's in there. So here we go again. I need to go to the 4 times. And here we go. More, and here we go again. Yeah. So that's basically the sand grains. And something that occasionally also looks kind of nice is if you try it in this here, which is dark field. Okay, so you now see that the sand grains are all bright, yeah, on a dark background. Okay, there's also something interesting moving in here. What is that? Okay. I lost it. Here it is. Almost looks like a I don't know, some kind of a worm, or I don't know. Yeah. Right now, right in the center, the thing that's kind of. Oh, it's interesting. Almost like a flatworm or so. Yeah, flatworm. Yeah, again, I cannot go higher, unfortunately, with the magnification, because I'm kind of worried, because I'm not using a cover glass now, right? So I'm a little bit worried of but maybe 20 times should work. Yep, still enough space. That's an interesting one. Yeah, possibly a flatworm. But you know what? I'm going to show you nevertheless. I'm gonna show that to you nevertheless, simply as a little demonstration. Because why not? And because I also would like to do that. So I'm going to put this away here. I'm going to do the following now. Put this away. A new one here, and again a little bit of sand. Then let's give it a try. What I want to show you. Now I'm going to take a little bit. I'm generous now with the sand. So what I'm going to do now is the following. You take a cover glass, and then I'm going to move on because I do want to show you the crystals as well. And then what you do is the following, because the sand is of course too thick, you do the following. You just push everything over like this, and you go like this, and you still some sand here, unfortunately. Not so good. Demonstration almost failed, but the idea is kind of to collect just the water here. Let's try this again. Let's try it again. You do that, you kind of push the sand itself to the side, and you just try to, this is better. Okay. And then the remaining sand goes back in here because I do want to keep it. And I just... Okay. And let's hope for the best now. Yeah, of course the sample is now not very dense, unfortunately. Yeah, of course. Yeah, yeah. Okay, here was something. This could be cyanobacteria. Almost certainly. Up, I bumped. Looks like it. Or not. I don't know. Yeah, it's moving. Could be, you know, probably cyanobacteria. Yeah. Yeah, in any case, you get the idea. And at this high magnification, unfortunately, there is not a, yeah. Let's try it like this. Unfortunately, there is not so much to see now. Yeah, some tiny things moving around. Yeah, I would say let's do the following. Let's move on a little bit, okay? I mean, yeah, I think you get the idea. Pond water in winter does have a few things to look at, and can keep you busy for the afternoon. Okay, so I'm just reading here a little bit, comments. Yes, okay. So what I'm going to do now is the following. I want to show you, yeah, as requested, I wanted to show you, I'm going to put this water sample away. I'm going to show you a couple of crystals, and we're going to quickly prepare also some citric acid. Last week I did a, let me put the slide away. Last week we did, how do you say, some vitamin C. I need to... And what I'm going to do now is the following. This is where I'm going to show you how to do some citric acid. And citric acid is very cheap. You can buy it pretty much almost in any store, because the supermarket. Why? Because it's used also for cooking. So you know, if you, yeah, also for cleaning surfaces and so on. So I got some citric acid here, right? What I've already done is I dissolved a little bit of citric acid in here, and I found out that it wasn't enough. So I'm going to add a little bit more citric acid now and dissolve it in some tap water, right? And that's pretty much it. Okay, and yeah, just so that you see what I've done. I did not wait or anything. I just put a few scoops in here. You allowed it to dissolve. If not everything dissolves, that's also fine. And then what you do is you have to essentially put it on a microscope slide and then wait for it until it dries. And because this is definitely too, how do you say, time consuming. For this reason, as you have over here, you can, you're not able to see it now because the cable isn't long enough. But I've got over here this, this coffee warmer where I'm going to simply put the slide on, okay? And it's going to take us a few minutes. And in the meantime, while this is actually doing that, I'm going to prepare a second slide of something. So let's put this one over here in first. So that's a little bit. Again, the thickness and concentration and the speed of crystallization, all of this can be varied. What I have done in the past as well, and this takes, however, quite a long time, is to directly heat melt the citric acid itself as well over a hot plate. However, if you overheat it too much, then it's not going to crystallize anymore. Yeah. So would heating the beaker while dissolving it be better? Yes, definitely. If you heat the beaker or warm the beaker, it's going to dissolve faster. Definitely. Okay. So so what I'm going to do is this one goes on here, right? And I'm going to wait. And the other thing that I wanted to show you is this one over here. This one over here, and I'm just going to, while it dries, I'm going to show you another slide that I prepared. Okay, and this one over here, which also kind of looks okay, especially in, this is now, I've got DIC using polarization, okay, and there are also some tiny interesting crystals here. It's kind of funny because I've tried this already before, so that is not citric acid, okay, this is not citric acid. Okay, but you see there are also these tiny crystals here, and let me turn the arrow, put the arrow away. Yeah, it's kind of interesting stuff here, and over here, some crystals are bigger, smaller. And honestly, I have here there are a few more. Honestly, I've seen nicer specimens. So maybe you're going to be more successful, but of course now you would like to know what this is. Okay. And here they're a little bit better over here maybe. Maybe let's go up and get more, 60 times. Yeah, so you see those tiny crystals here. Yeah. So I'm going to now show you what this actually is, okay? And I know that you're gonna think I'm totally crazy now, but yeah. Sorry guys, it's gotta be. This is the thing that I've got here, and this is that's urine. I'll just say it as it is. It's, in other words, it's my own pee, and urine makes beautiful crystals. It contains urea. And and what I have done basically is the following. I simply have taken a little bit of urine, put it on the slide and dried it. And that's it. Okay. The idea is the following, that there is, of course, urea in there. I think there's some kind of phosphates as well. Depending on the drying time, I actually had some nicer samples as well. But it's really that easy. Yeah. So what I've done is the following. If you look at urine, this one is actually a little more clear. It is possible to centrifuge the urine. I tried it as well. And urine basically should be free of bacteria, but there are still body cells in there because of the epithelium. Yeah, I was able to see a couple of body cells as well. And what I've done today in the morning is I did a little bit of staining. And just to show you, and then I said this, I'm not going to show you, it's not very interesting. But I'm going to show you in any case. Okay. Let me turn this down. So that is now basically a centrifuged urea. And I've done a crystal violet staining, and all of these things that you see over here, those flat things here, these are cells, basically that have come off. Yeah, so this is, so those flat things that you see here, these are basically cells. Sometimes you can even see a nucleus inside them. So these are epithelial cells that basically came off, and I stained it with crystal violet. Yeah, those fibers that you see here, that's basically contaminants that were on the slide. Yeah. So in other words, urine is interesting, therefore, for multiple reasons. Because it contains cells from the body, and of course because it makes nice crystals. Yeah. So that's basically the cells, and if you just look at them, you can actually see that many of them in here, the tiny dot, that's the nucleus. So if you want, you can go up a little bit with the magnification. Let me take this away here. So that's now again in bright field. So you see the epithelial cells here. So they look kind of very similar to the cheek cells that you take. So just showing that there are cells in urine as well, and basically I put this over there, and the crystals there that I just showed you, where I don't even remember where I put them. So, and right now I still see that I think I have to go up a little bit with the heat here, because the citric acid is not ready yet. Where is the desk view again? Okay. Yeah. So basically what you can do is if you take a little bit, a small drop of the urine, it goes on the slide. And the thing is the following, I know some people feel disgusted by this. Let me assure you that actually fresh urine is fairly clean. Yeah. It will only start to grow bacteria when it turns bad. Okay. So just saying, it's I know psychologically it might not be the nicest thing to think about, but actually fresh urine is fairly clean. Yeah. So I'm also going to put the urine slide on here. Yeah. And you cannot see that because here it is. Yeah. It's the urine slide, and that's the citric acid slide. Yeah. So, and the citric acid slowly starts to be finished. And another thing that I prepared, we're just going to have you used meshes of different densities? I need to disappear beneath my desk. Let me just a second. So I'm back again. I am back again. Oh man, meshes. You want to see a mesh? Let me show you what I organized. What is this here? This is a so-called a plankton net. Which I have not tried out yet. Okay, so that's basically something that is on my list. Yeah. So that's simply something that I would like to do as well. So you pull it through water, and there is a very fine mesh, which I can put under my stereo microscope maybe. And this will basically then be used to collect algae and so on. And then you can collect it in here, and then you can do some kind of a sampling. Yeah. So that is indeed something that, just because I just wanted to show it to you as well. Yeah. So next thing is the following. I burned myself. Okay. It's so hot. So let me see. So here it is. I need to turn off the. So very quickly. This here is now the slide with the citric acid. You see, well, it doesn't really appear like to have many crystals here. However, crystals will actually start to slowly form. So citric acid, the crystallization of citric acid is not quite as fast. Okay, so we have to be a little bit patient here, but I'm going to show it to you in any case. Yeah. So let's have a look here. And then let's put the scope view again. The arrow goes off. And we have to find somewhere a crystallization start. Okay, because right now we don't see a lot. But sometimes here we're now at the edge here, and then sometimes what we might be able to observe, and we have to be very lucky, we might be actually see the start of a crystallization process. And if this doesn't work, then we can actually try to seed it a little bit by touching the surface. But you see over here, not a lot of crystallization happening. Okay, so we're just going to wait a little bit longer. A little bit of dirt is fine, because if there's a little bit of dirt present, then this can act as a crystallization starter. And because I knew that you wanted to see something, I'm just going to show you now a slide that I prepared before. Okay, so that is, for example, citric acid, yeah, which I prepared just before. Yeah, you can actually see that, yeah, there are these nice patterns. This one over here, a little bit like snowflakes. Yeah, I think it's also quite pretty. Yeah, but we're going to see whether the other slide will start to crystallize as well. Yeah, we can actually go in a little bit more here. I think yeah, a little bit artistic. So that's basically also one of those images I also used as a thumbnail for this live stream. Yeah, yeah. So let's have a look again over here on the other slide. So I'm just going to touch this a little bit. I'm going to show you here. Okay. Sometimes if you touch it a little bit like this, sometimes this acts as a starting point for crystallization. Okay. So let's have a look here. Yep, here we go. Okay. So that's basically the thing where I touched it. And yeah, you can now see some crystals forming here. Okay. So this is the place where the citric acid was very sticky, right? Basically by touching it, I started the crystallization process. And if we are very patient, we can actually see them grow. But again, it's a little bit slower than, or quite a bit slower than, for example, with the vitamin C of last week. But if you're very careful, you can actually see how the crystal is actually growing. Yeah. Yeah. Yeah, I'm just going to leave it as it is right now. And you can also check some of the other crystals that you see. Yeah. Yeah, over here, 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 here. And you can actually see how the edge over here is kind of, yeah. Also starting over here, it's going to reach the side of the, video. It looks kind of pretty. Yeah, if you want to try this yourself, very easy. You need just polarization filters, 2 of them. Yeah. Actually, this is also quite nice. Yeah. So again, you evaporate the water, and it will start to form this sticky surface. And then you have to kind of wait until it starts to crystallize. And sometimes you simply, or you can add a little bit of citric acid powder also to start the crystallization process. Yeah. Let me go down a little bit with the magnification again so that we get a better overview here as well. Yeah. Looks pretty. Yeah. And over here, no crystallization yet. Maybe they will start here. Yeah, citric acid likes to fan out. Yeah. Won't the water vapor fog up the front lens? No, because the water has already been evaporated. So basically, what I've done is I've put the slide onto how do you say, on a hot plate to evaporate the water, and then what was left over was a sticky, syrupy layer, which might still have contained a little bit of water. But it's not warm anymore, right? It's the slide has already cooled out, yeah. And I'm watching this through a loop lens, and the red bits of the crystals start to pop out in 3D. You mean those ones that you see over here? Yeah. I mean, yeah. First of all, the crystals are three-dimensional. I'm going to now remove the polarization filter. And this is how it looks without, without polarization. Yeah. I mean, I do have DIC, but honestly, it will look similar. It will look very similar just in cross-polarized. I could actually try that. I have to do the following. I'm going to remove now the top polarizer. I'm going to put in the bottom one. I have to use, however, the how do you say, the lowest magnification, and I'm going to now add over here. I'm going to add now a polarization filter, if I find it. Where in the world did I put it? Here they are. Here it is. I'm just going to drop one over here, directly on the... Here we go. And, look, it's actually... no color, but it also looks kind of nice. Yeah. So that's now just using a regular polarization. Yeah. You have to rotate, one of them that actually becomes totally dark. Yeah. And basically they will start to continue to grow. What I'm going to do now is I'm just going to show you I'm going to show you now also another way how you can seed the crystallization process. That is by simply doing the following. I need to always switch over back to the desk view. So I'm going to turn off I've got the light here. Yeah, you can actually, here you can actually see it much better. Let me refocus this, if I can get into focus at all. Here we go. You can see those white parts here. That's where it starts to crystallize, right? And you see the rest here is looks sticky, syrupy. And what you can try to do is the following: you take one of the citric acids crystals, the non-dissolved ones. Just take one of those grains here. And what you do is I like to do this as well. You just take this here, and you just basically drop it on here a little bit, like this. And this way you're seeding it as well. So I kind of hope that the crystallization process starts here, because some of the crystals broke off, and maybe we're able to see something here. And then you can do a time lapse. I'm going to just use and I need to refocus again. Yeah, I lost the... Yeah, you get the idea. Oh, I get it. I have to do this here. Here we go, a little bit more light. Yeah. So this is again, now this is the place where I actually scratched over it. You see this line here? This is kind of where I dragged the crystal over, And this is where the crystallization started. It's pretty fast actually. Yeah. And you see again over here, You should be able to see how it starts to, yeah, grow. Yeah, it's pretty nice. So in the past, I've made lots of pictures like this, and then you can hang them up on the wall and things like this. So that's actually one of the easier ways. Again, what I've got DIC, so but I have gotten extremely colorful images, also just with regular crossed polarization. So most of you, or pretty much a lot of you, don't have DIC. You don't need it. You can get a lot of colorful images just with regular crossed polarization, right? Have you ever tried heating the solution and getting it super saturated and dropping a single drop on the cold slide for instant crystals? I have not tried that. What I have tried, however, is the following. I'm going to actually, I'm just going to leave this beautiful picture, but what you can try to do, but it does take a couple of, maybe a day or so, yeah, and that's basically the following. I just show you over here. You put one crystal from the powder on the slide. You put a cover glass on top, which of course is not going to be flat because there's this large grain in between, and then you put the whole thing on a hot plate, and you're going to melt the crystal, and then the citric acid will kind of, when it melts, will make a very thin layer between the cover glass and the slide. Yeah. And then you can wait for the crystals to form. However, this takes about a day or so, right? And if you heat it too much, it's not going to work, right? So you just you have to be careful that you don't heat it too much. But essentially, I've already melted citric acid directly on a slide over a hot plate, and this also gives beautiful crystals. Yeah. However, I find this method with the water and evaporating the water goes faster. Yeah. So. But they're different. I just want to say they're different. Different ways. Look how this one is growing already. Yeah, it's pretty cool. Yeah, I mean, I've been doing this so many times now, but I'm still always fascinated. It's just very pretty. Yeah, yeah. So. I use my finger to spread a thin layer of liquid soap on the slide first. Then the solution easily spreads across the slide because it breaks the surface tension. Evaporation of the solution is faster. I don't need a warmer. Yeah, that's an interesting one. I did not try that. So very briefly, the problem is the following. The problem is that when you put a drop of liquid on here and it evaporates, thank you very much. And it evaporates, then essentially it will start to, because of the surface tension, will start to become smaller. You want to have a thin layer. And one way of doing that by breaking the surface tensions, as it's mentioned in the chat, is by putting a little bit of liquid soap on the surface. I did not try that yet. Okay, and then when you put the citric acid or whatever solution on it, it kind of spreads nicely, and then you get an even thin layer, and then it also will evaporate faster. Thank you for the recommendation. I did not, how do you say, try that yet? Yeah. Yes. Yeah. If you have a piece of plastic, CD case or similar, well, not a CD case, but this was also a request to put some tape, on, under the microscope. So what I'm going to do now is this is actually also something that I would like to demonstrate here. And I'm going to click away this here, and this is also something that you might want to do. Look, you can use the following. You can use those filters that I have here, and if you don't have them, then you can also try to use something like this. Okay, yeah, they are also polarizing. Or if you don't have those, you can also try to use something like this. Just a second. And I know I'm kind of repeating myself from last week, but some of you might be new to the live stream. But you can also buy those thin polarization filters as well. Yeah, they have a plastic, how do you say, protective cover here. But they also work, they're pretty cheap actually. And these are these are circular polarizing, so the orientation here is also important. But I've also experimented with those. And what I've done here is I put some sticky tape on here. And I scratched the surface a little bit of the tape, and then you put another polarizer on top, like this. Yeah, and I'm going to show you this now under the stereo microscope because this also looks kind of nice. And we've got this here. I need to turn on the stereo microscope. Here we go. The arrow goes away. And I'm going to zoom out a little bit here. And yeah, and when you turn, look what happens when you turn the top filter. You get those nice patterns. And if you want to know what these, how do you see these round lines are, I'm going to show you how you do that. You take a very sharp and pointed object, like my tweezers, and what you do is I remove now the top filter. Okay. And what you do is you just scratch the surface of the plastic. Okay, and then you get something like this. Okay, so you actually see that, yeah. And the interesting thing is, if you turn it, you see that, yeah. And the reason why sticky tape is polarizing is because apparently the tape, when it was manufactured, was stretched, so the plastic molecules are oriented in a certain direction, and therefore you're able to get this polarization effect. And here I've got some more tape, and I'm just going to show this to you. Okay, so I'm going to add more tape over it, and you won't see a lot now, but again, when you put something, yeah, you can actually see how this changes. Yeah. So this works quite well with sticky tape. Yeah. Just saying, there are plenty of artistic things that you can do. Yeah. Yeah. So yeah, so that's also a little bit because has also been a little bit of a request. If you go to the beginning of the chat, somebody asked if I can put some tape. Yeah. Yeah, on here as well. For secret messages, yes. Yeah. Yeah. So yeah. So there are plenty of things to play around with. But actually, if you actually zoom in quite a bit, okay, you can actually see it like this. Yeah. Yeah. So just wanted to demonstrate this as well. And we move on a little bit. What else did I want to show you? There has also been another request, and the other request was to put some washing from the washing machine, some water. So I did collect a little bit of water from my washing machine, and it's again not the first time that I'm doing this, so I do know what to expect a little bit. Yeah. And this is actually something where sometimes some people are a little bit worried about. Just want to show this to you. It's nothing out of the ordinary, nothing that we don't already know, but there is a concern that people have. Okay, so this is basically from a washing machine. There is this hose, this tube that you can open, and you can let out the water. And this is kind of the water from the washing machine. Now I'm just going to take some of this stuff here, put it on a slide. Okay, and cover glass goes on top. Let me put this away. Again, I have to always be careful that I don't, how do you say, dump or bump and spill everything. Cover glass goes on top. And now this is something that I would like to show you again. This has also been one of the requests, and I thought that's actually a good point, because this is something that maybe I hope that I'm able to show that to you, the thing that I'm kind of worried about. I have lots of dirt and so on. Yeah, but we already see the worrisome thing here. I don't know, not identifiable, many of these things, but there is already something that is of concern. Yeah, of course. Yeah, all of those fibers from the clothing, lots, if they are synthetic, lots of microplastics. Okay, I'm just saying this is indeed one of the things that people are really worried about. That they're, because of that, the runoff of the water, those microplastic fibers essentially cannot be broken down, even if it goes to the water cleaning facility. Yeah, these are synthetic fibers. This basically means they cannot be broken down because in nature there are no enzymes. Bacteria and so on, they don't have the enzymes to break down microplastics. Therefore, they're not decomposable and they end up in the environment. Right, and that is essentially apparently clothing is one of is a huge source of those fibers. Okay, so this is just something that I want to mention because also for your info, yeah, recently I got yeah, emails and where people find them under the microscope and they kind of wonder, are these worms or what are they? And no, these are not worms, these are just fibers. Many of them could be cellulose fibers from cotton or so, but some of them are actually stained, like the ones that you see here. You find a lot of blue ones as well. Let's put away. So that's again bright field, right? So you can actually see that there are also blue ones around there, right? And many of them are actually indeed synthetic fibers. Yeah, and yeah, that's of course yeah, cause for concern because yeah, they can be found in the environment, and honestly, I find a lot of them also in water samples and so on. Yeah. So having said that, if you feel that this looks a little bit ugly, this mess, then I do have a slightly nicer specimen to show you. And I'm going. This is also today. I'm showing some weird things. Look at this one over here. You see this fuzz over here? Yeah, isn't this beautiful? Lots of fibers. So I'm gonna put this also on a slide. You now want to know where do I have those from? How many of you can guess? See? No, this is not house dust. This is not house dust. You know what? I'm going to add a little bit of water indeed. So I need some water. From over here. This is not house dust. This is basically, yeah. That's a fuzz from my belly button. It's something that's quite well known. Is that yeah, over the course of several hours, yeah, there is kind of the clothing that you wear, those fibers that rub off, and they will collect inside your belly button, and this is basically some of this fuzz. And it kind of gives you a very nice overview and idea of what actually is present in our clothing. Yeah, so cover glass goes on top here again. Okay, and yeah, this is just basically the fuzz from a few hours. Yeah, and that's what we get. I mean, yeah, and of course, if the t-shirts and the clothing that we wear contains synthetic materials, here it is. Okay, so yeah, that's just saying. Actually, normally I think in dark field it looks also kind of pretty. Yeah, let's try it in dark field. Yeah, I have to kind of open it a little bit more. So here we go. It's a little bit overexposed, maybe even. Yeah. Yeah. So look at this. I mean, yeah. So not surprising if a lot of these fibers end up in the environment. Again, I'm not so much worried about cotton and cellulose fibers and all these things that they're going to be broken down, right? That's not the issue. It's more the synthetic ones that basically is, yeah, microplastics. Yeah. Yeah, you can. That's actually a nice thing. How long does it take for this stuff to build up? Yeah. So yeah, just wanted to show this to you as well. So sometimes yeah, they also can look pretty, but actually they're not a good sample to have in the environment too much. So let me quickly check. Ooh, 1 hour 15 already. You know what I'm gonna do? I'm going to very quickly read through the questions here. Michael, my son, who's been preparing those questions and copy pasting them from the chat, told me today that this is the 100th page. Okay. Yeah, so a phone display maybe next time. I'm running a little bit. There has been a request to put a phone display. This sticky tape experiment. Insects under the microscope. Answered. So most of the questions are already answered actually. Do you have a Discord or other discussion forum? And yes, I do have one. If you go on my webpage, microbehunter.com, it's called the Microbehunter Microscopy Forum. However, and that's important, you need to send me an email. You need to register and sign up and send me an email. And then every couple of days I will go through my emails and I will basically unlock you. The reason is in the past we've had so many spam registrations that this was a real problem. So not Discord, but it's a discussion forum. I mean, if you want to, it's not very active. I also have a Reddit page where I post my videos. Yeah. I often see clusters of diatoms. Is this a nursery or are they all biting on the same Mars bar? Basically, very often you have them in colonies. You've got one diatom, it's dividing. Yeah, then you've got two of them. And because many diatoms are sticky, they form a sticky layer, so they basically form some like a colony. Yeah. Have you tried using mashes of different densities to isolate microorganisms from the rest? Yeah, I just showed you one, which I might put under the microscope as well. Yeah, the dish soap, I already talked about this. Yes, okay. So very quickly, let's have a look at this mesh here under the stereo microscope. Because this is, and it's again also something that I'd like to try. Yeah, so, okay. And you see, it is a pretty fine mesh. I mean, actually, I should be able to measure it out. Yeah, but this is a, I'm now already at maximum magnification here, right? So it's a fairly fine mesh. It's a plankton net. So forget about nylon stockings and so on. Yeah, not fine enough. This is really fine. Yeah. So yeah, I did once register Discord. Honestly, I don't know much about Discord, but I have to be a little bit careful that I'm not spreading myself too much because I've got a YouTube channel, I've got a web page and the forum, and if I've got too many things going on, then it's difficult for me to manage all of them. But if you look at this plankton net, it's indeed very fine, and I'm not able to see the mesh. It's almost like cloth. Yeah. So last but not least, I do want to show you, or not show you, because I don't know if there is a lot to see or not. Let me clean off the slides a little bit here, because I've got too many slides floating around. I do want to put a sample of an enrichment culture under the microscope, which I've done some time ago. So this is a water sample that I have from, from one of my jars, and I basically added some how do you say, those white things that you see in there, that's oats, some cereals. This is now over a week old, or 2 weeks almost, and it looks pretty disgusting. This basically means that there are plenty of bacteria here, and I don't know if also some paramecia started to grow. If you overfeed it, then bacteria will start to explode. They will take away the oxygen, and also the paramecia not going to grow. So I don't know how this has developed now over the last couple of days. So I'm gonna try that. Okay. Yeah, you want to be a little bit careful with these things, yeah, right? Because bacteria, so open wounds or anything, yeah. That's why I go in here and I try to grab a little bit of whatever I've got here. The tip immediately is being disposed of. And then what happens later on is, of course, I will disinfect the whole thing. Because, yeah, cover glass. Here we go. And it's a little bit out of focus, I just see. And this goes also away. So all of the slides go there. And then usually what I do is when I do wash my slides, I usually pour boiling hot water over them. And scope view. And let's have a look again here. Yeah, I mean there are. These are rather yeah. Here, yeah, there are some seem to be paramecium here. They did not. Couldn't are they? No, there's not paramecium, but it doesn't matter. Some ciliates. In any case, again, those fibers everywhere. However, we're gonna go up a little bit with the magnification because I'm now using a low power. Yeah, okay. See, because they're kind of feeding on the bacteria, of course, and then they're starting to multiply. And I was some time ago, I was very lucky and I had a very high concentration of ciliates. Yeah, and yeah, here there are not so many. Let's go up yet further. Yep, and I'm going to do the following now. I'm going to show you some phase contrast because I have actually seen some spiral-shaped bacteria as well. Yeah, up here we go. Yeah. So those tiny things that you see in the background, where is this? Those tiny dots, those things here, yeah, these are all bacteria, of course. And there are plenty of them. That's why yeah, very high bacterial density. Okay, the washing machine sample, you would like to see it also in phase contrast. Yeah, I'll do that. Yeah. So just for your info, that is basically this was a water sample where I added some cereals, okay? And then you see that yeah, some of the organisms started to reproduce a little bit more. Okay, so however, I don't know about so I'll do that because it was you requested. I also put some how do you say that, water sample from the washing machine. However, I'm not quite sure if there are a lot of bacteria in there or not, because of course with a how do you say washing machine which is boiling hot. But then again, let's have a look. I'm going to take a small water sample directly. So yeah, so I've just made this from the washing machine. I just check the time. I slowly have to stop again. So yeah, I just prepared again from this is a slide that I just made from the water sample from the washing machine. Okay, no solid materials, and let's have a look now. I'm just gonna move over, and here we are. No, that's not. That's some dirt. Okay. It looks. Yeah, I think the density is a little too low here. Could be that those tiny dots. Yeah. I think what I have to do, and maybe that's something I'm gonna try doing, is I'm going to maybe, I'm just going to centrifuge that. You know what I mean? You do see a little bit of movement. I don't know if you're able to see it I'm able to, yeah. There are some bacteria moving around. I don't know if you're able to see that. Yeah, but as you can see, the bacterial density is significantly lower here. Yeah. So I have to probably centrifuge it, and then I'm able to see a little bit better. Yeah. Why are you always in such a hurry to stay on schedule? That's a good question. Because I do know that some people basically also want to go to bed because they do have, yeah, because of the time difference, it's in the middle of the night. So, because he has a life outside of YouTube, I don't know so much. No, I genuinely I like how do you say I like doing videos, of course, and I mean ultimately you already know that I am a teacher after all, so I like to share my experience. So let's go back again to the enrichment culture. You see, if you look very careful, you can see that there are some spiral bacteria as well, where you want to be a little bit careful because some of the spiral bacteria are not quite healthy. Yeah? Wow, quite a lot going on here. We want to sit all night and watch with you. Yeah, and something over here that's also quite interesting to observe. You also occasionally see that. See, sometimes you can actually see that there are bacteria clustered together, so it looks almost like a microcolony. Yeah. You still never responded to my question in my last email. Are you talking about the spermatozoa? Yeah, honestly, if you wanna send me, yeah, if you've got some nice videos, sure, please go ahead. Okay. Please, Danelle, yeah. So. So I'll quickly check how do you say, I'll quickly check if there are any questions. Yeah, for me, not so many here. Phase contrast. Write your name. I'm a little bit, yeah, I don't know what this was actually referred to. Write your name. I'm Oliver. If this was kind of, yeah. Have you tried heating the solution and getting it in super saturated? Yeah, I have not tried that. Yeah, and then the dish soap thing. Yeah, we tried that. Yeah. Oh, write your name on the tape in scratching. I see. That's the thing. Yeah, that would be a little difficult because I don't know if I'm able to write that small because the microscope does magnify quite a bit. Yeah. So yeah, so that's more or less all. Just let me check if I wanted to show you something else here. The sample over here. I've got some other water samples. I'm not going to show everything to you because I do need something also for the next couple of weeks. Yeah. Shout micro and arranged diatom slide. If you have some arranged diatom slides, they're pretty expensive. Honestly, I tried to get one. Share a picture, please. Yeah, arranged diatom slides were quite popular back in the day in the 19th century, where people actually really made very beautiful patterns by arranging the individual diatom shells on a microscope slide. I have no idea how they have actually done that. They've apparently used eyelashes. And yeah, to move them around, they mounted an eyelash on a little stick, right? And then they were kind of moving around the diatoms. But honestly, I don't know how in the world they were able to keep it like that when they added the mounting medium. They must have glued it on somehow. I don't know how they've done that. Yeah. Yeah. So yeah, I have some arranged slides, but nothing fancy yet. They go so expensive. One went for eBay for £250. Yeah, So that's quite something. Yeah. So these are quite beautiful slides, but yeah. One of just maybe before I stop, there's one thing that I just also want to share, or a thought, and that is, back in the day, even back in the 19th century, people really did some highly artistic slides and so on. They were polishing down and grinding things like crystals and so on. Sometimes I feel we have to be careful that all of this knowledge does not get lost somehow, right? Because sometimes I wonder, how did they actually do that, right? And then so I'm just saying, let's all take care that the knowledge and the experience that the people had in the past, that somehow it does not become lost. Yeah. So yeah. I can't even afford it for myself. Yeah. Yeah. So I'm going to do the following right now. I'm going to call it a day for today again. No, I'm not yet. Not yet, because I'm going to try something nasty now as a last act of today. So I'm going to kill those bacteria using some alcohol. Why not be nasty a little bit? So I'm going to put some alcohol here. So this is just, I'm going to drop a little bit of alcohol right next to the slide and see what happens. So I dropped some alcohol in there. Maybe I need more. It didn't quite diffuse yet. Let me see. Ha! What this? Let me check. Did it reach? Did it reach? Did it reach the cells or not? I added a little bit of alcohol to the edge here. They don't seem to mind. Seriously? I mean, this is a little surprising. Ah yeah, here they're dead. Here, not some. Someone's still moving. Wow, no, this cannot be. Nah, I think the alcohol has not reached them yet. Let's do it like this. This is now a slightly lower magnification. Okay, I think, yeah, I think I have a slight diffusion problem here, because over here the alcohol has maybe not completely mixed in. Over here, yes, but over here, yeah, apparently the alcohol has not reached those cells yet. Ah, but they seem to be moving away. That's why they're over here. See? Maybe they're sensing they're approaching alcohol somehow, and that's why they're moving away. And that's why they have accumulated over here. This could be it. Yeah. Yeah. Yeah, just saying. Yeah, I think I'm just going to, I'm going to call it a day for today. Yeah, I wish you all of you all the best. I think I might see you again hopefully next week in the new year. Wish you a nice transition into the new year, and I'll do the following. If you have any recommendations of what you want to have a look at, please write it in the chat again at the beginning, and we're gonna take it from there. Yeah, again, have a few nice, in case you still have holidays, a few nice days off, and all the best, and hope to see you again. Bye bye.